Hi, Paul. Right. Welcome. So welcome. If you are joining me, I am over on my YouTube channel with this screen and going live. It's got the Zoom link there too if you want to join. I did say I would look at some paper 3 questions today. So yeah, I am on Zoom today. It is working for me today, thankfully. I think it was just having a bit of a bad day, I would say, on Wednesday last week. Okay, so I'm going to get rid of that. And yeah, welcome if you are coming along and joining me. So what I did say to those who were here on yesterday's session actually is that I would do a bit of paper 3. So if you are joining me for the first time and you're interested in covering some questions, then please ask away. I will jump between the 2 screens to try and answer any questions that you may have. But I am going to start with the paper 3 content because I did say that I would cover that in today's session. So we're going to carry on with the 2019 paper because I do think this is a good one to show you how they can link things together with the paper 3 content being all of the modules. So what we want to do then is look at the questions and make our way through. So here we can see we have got information about the naked mole rat. So if you are just joining me on TikTok, guys, I am on my YouTube channel and there is a link to the Zoom live lesson if you want to come along. And we are going to look at paper 2019 paper 3. So if you do want to join me, then please do come across. So, and obviously if you have questions as I go, please do ask away and I will try and answer any that you ask as we are going. So here we have a question, which is looking at this naked mole rat. It's really hard to say that a number of times. And what are they telling us? Now, it's really important that you highlight information that they've told you in the question. So we have a naked mole rat that they've told us about. Can I go from a C to a high B starting from now, covering 4 topics a week? Ghost, yes, I do believe that you can go from a C to a B as long as you are actually focusing your revision. There is plenty of time to jump up at least 1 to 2 levels in the remaining time, but as long as you are honing in your knowledge. Amna, yes, absolutely. Let me jump across then. You want me to go through question 10 on paper 2017? Let me just see if I've still got that one open. And you're actually here on the session. Let me just go to 2017 because we have been doing 2017 before. Actually, maybe I don't know if that one opened. Okay, let me open it now and let me bring it up. So 2017, question 10, paper 1. Here we go. Paper 1, 2017. I want to open that with this one here. Okay, so let's have a look. You said question, I did think we've been doing this one. I've obviously been looking at it somewhere. Question 10. This one here. This is the question, right? That you want to cover? Yeah. Okay, so this is an interesting one. I actually really like this question. Again, because it says to you that you have got your hormone. So you're being given a hint in the question, multiple choice question. The hormone is synthesized in this prothoracic gland found in the upper thorax of some invertebrates. Whoa, hold off a second. They're talking about invertebrates. We've never done invertebrates. We have only got, good luck, Alex, in your mock tomorrow. We've only ever covered the invertebrates when we look at the gas exchange. We've only ever covered the invertebrates when we talk about the circulatory system, and now they've brought in hormones. So this question does throw a lot of students off, but actually it is important to note that they've told us the information here. So they've given us information that it's a hormone. They've said that it's going to be released from a gland, and that gland is found in the upper thorax of the invertebrate. It's going to be released into the hemolymph. So just like our hormonal system releases our hormones into our bloodstream, and it will then be transported to the cells, the target cells. So here we have that the cells near the surface of the body, and that then causes the loss of this exoskeleton. So that's the response, that's the signal, the chemical messenger, the hormone that's being released, and now what we've got is it transported in the bloodstream, and it is then going to reach the target cells, and it will cause the target cells to do whatever needs to happen. So I've not even looked at the information yet. They've given me a lot of clues in that first part of the question. So it's always important when you are doing this that you take the time to, oh, give me 1 second. I've just put that down. Not mean to do that. Paused on me. I've got too many screens open, that's the problem. It's now saying, we're not interested today. Try, there we go. So yeah, it's given us some information here to say, right, well, what do you actually know? So do you understand that the hormone that they've told you is synthesized by specialized neurosecretory cells? So first of all, I'm going to highlight this and say, well, what is a neurosecretory cell? What does that actually mean when I talk about a neurosecretory cell? Well, a neurosecretory cell is a cell which is a neuron that is linked to a gland. So it's a neuron linked to a gland. And that neuron that's linked to the gland is going to result in the secretion. Instead of a neurotransmitter, what's going to happen is this is going to secrete a hormone. And you're going to sit there and go, well, I've never been taught about this. My teacher hasn't mentioned this at all. So therefore, I am not in a good place when it comes to this topic area. But what the question actually says is, it says, which of the following statements explains how this hormone is able to act on the cells near the surface of the body? It's not asking you about how this hormone is released. It's about how it's actually able to act on the cells in the body. So therefore, let's have a look at the rest of the information and see what they tell us there. The exam board is at OCR for those of you who are joining me on TikTok. I am on my YouTube channel, so if you want to jump over, you'll be able to see the screen, see the question, and ask questions there as well. You're doing advanced higher biology. It is similar, but there are some differences because you've got different exam boards, and the Scottish advanced hires are slightly different. But again, it just depends which exam board you're looking at with regards to that one. So let's go back and look at some more information. So we know it's a hormone. We know it's a hormone that's being released into the hemolymph, and the hemolymph is basically the blood circulatory system for the invertebrates. So therefore, this hormone needs to be soluble in the hemolymph, and the reason it's soluble is because it is a polar molecule. So we know that this one is going to be correct because they've given us the hint. We know that our hormones need to dissolve in our bloodstream, and we know that those hormones are then going to be transported, and the reason they can dissolve is because they're soluble, and that solubility is to do with the fact that they are polar molecules. So then we need to look at, well, what else have we got? So this hormone is complementary to the cell surface receptors on cells throughout the body of some invertebrates. Well, they told us that it's transported to cells near the surface of the body and causes the loss of the exoskeleton. So we know that whatever we've got as a hormone, that hormone is going to bind to a receptor. And because we've got this hormone and a receptor together, if they bind together, what we end up with is a hormone receptor complex. Hormone receptor complex. And therefore, they need to be complementary for that binding to occur. So because of that, what that therefore means is that this one is also correct. And we know that it says explains how it's able to act on the cells near the surface of the body, not how it is released. Therefore, we wouldn't agree with number one, because that's about the releasing of it and where it's made. It's actually looking for you to say C is the answer. So does that help with how to answer that question? So guys, if you are just joining me, I am on my YouTube channel, so please do come across. Great, but if you have questions like that, please do ask away, because that's what these sessions are about. They are for me to help you. I'm not planning them. Obviously, it's not like my group tuition sessions or my one-to-ones where I plan them meticulously. If you have a question, ask away, but hopefully I can then help and guide you on how to answer them. So do I do predictions? Not really, but what you'll tend to find, Greg, is that I will maybe say something that pops into my head as I'm doing it. Yes, I am answering questions on my YouTube channel. So Biology with Christine, jump over. You can actually join the live session that I'm doing on Zoom as well, if you would like, because the link is there. So let me just double check. This is what I forget to do. Right, here we go. There we go. Right, so looking at this question then. So we've got a naked mole rat is a mammal. However, it's several features that are unusual for mammals. So straight away, the fact that they've told us it's a mammal is the hint. And I would say to you, when you're going through these questions, look for these hints. So there's my hint to begin with, that it's a mammal. And I know that they're going to be looking at, because they're talking about regulating in this next part, the naked mole rat regulates their temperature. So I'm just going to make a little note to myself that that's a hint to me. And I know that mammals are endotherms. So if I know that mammals are endotherms, I have to think about that in the best way I can to try and start to bring forward some of my knowledge. So let's go on and have a look at what they are telling us with regards to this question. So we have got some features of thermoregulation in naked mole rats are listed below. So they live in complex underground tunnels, which tend to have a stable temperature. So they're in underground tunnel systems. We have a stable temperature between 30 and 32 degrees C. However, sometimes the environmental temperatures can increase or decrease outside this range, okay? So that's important for us to note. In experiments that examine environmental temperature changes, the core body temperature of the naked mole rat remains close to that of the environmental temperature. Environmental temperature, that's normally what an ectotherm does. So this is slightly different than the endotherm that we're expecting, because they've already told us that they're mammals, but they have told us that they have some unusual features which are unlike mammals. So that's the bit we need to be looking at together. And then what we need to say is, okay, so these tunnels, temperature increases, they often move to a cooler part. So they're going to move to a cooler part. So we know that if you move somewhere to that cooler part, yes, let me just finish this one question, then I'll jump into what you're interested in. So when they move to that cooler part, that is a behavioral response. And that, again, is something that we'd expect of a ectotherm, more of an ectotherm than we would of an endotherm. Now, It doesn't mean that we don't do these sorts of things with regards to being an endotherm. Like earlier, I got really hot so that I took off my hoodie. Well, after I had done what I needed to do, I then actually felt a bit of a chill. So I got cold, I put my hoodie back on. Now, my physiological responses that occurred, i.e., things like, when I get really hot, you'll see it, I'll start to go a bit red. You'll see my cheeks will get really rosy. That's also because I get a bit excited and over exaggerated when I'm teaching. But that's actually a physiological response. What you can have when you get cold is the fact that your hairs will stand up because your erector muscles are going to contract, causing your hairs to stand up, so you get a layer of air. So these are things that we would expect of endotherms, and therefore they're showing us these behavioral responses as well. So let's just carry on and say what they do. So when the tunnel temperature decreases, they often lie together in large groups. So if they're lying together in large groups, they're huddling. And therefore, they're trying to share the thermal energy amongst themselves. So when you think about this, you know that thermal radiation can result in the loss and gain of heat. Well, so can conduction. And then you've also got convection, and you have the evaporation side of things. So what you have to understand is there are hints here in the question. They've given you that information, so try to use it in your example. So outline 2 ways in which the thermoregulation is different from the thermoregulation in most mammals. So basically, use this information that we've told you in the question and actually apply what you know about the fact that mammals are endotherms. So the fact that they're naked, what do they not have? Well, when I talked earlier, I said about the fact that my hairs are going to stand up because of the fact that my piloerectal muscles are going to contract, causing the hairs to stand up so that I get a trapping of a layer of air. These are naked mole rats. They do not have that hair because they're naked, and therefore they're not going to be able to respond in the same way that we would. So there's no fur, no hair to trap that layer of air. Therefore, what do they do instead? They're going to use behavioral responses like huddling to keep them together, to keep that warmth, because they will adapt to the environmental temperatures. They told us that already in the question, so we need to make sure we're using the questions as a way of explaining it. So behavioral responses are what we'd expect mainly to be used in ectotherms, whereas endotherms, that would be more the physiological responses. Do you know any resources for those OCR A biology practicals in exam context? So Joyce, every single video I've got on my YouTube channel, I've incorporated the practical skills as I've gone. I have done a workbook specifically on module 1 content, and that therefore covers hints and clues on how to do it. And I haven't done it specifically to the practicals which are within the specification module 2 to 6 because they're in my videos already, and I'm still building the other workbooks. So I would say definitely have a look at my YouTube channel, but they're incorporated wherever there is a part of the specification. So if it says, for example, dissection, I've talked about the heart, I'll show you a bit of the dissection with the lungs, etc. There are little bits through all of it. Okay, right. What were we doing? Lynn, you asked me for 21A on paper 1, and we're looking at 2018. So 2018, paper 1. Let me just open this one up. So where are we? 2018. Paper 1, and you said 21 AI. Where are you? 21. 21 AI. Okay. So you gotta love a question. Full papers for A-level biology, you need to be doing them now. You need to be doing as many as you possibly can now, and doing paper 1, paper 2, and paper 3, not just focusing on paper 1 because it's the first one, then paper 2, then paper 3. That won't give you enough time. Paper 3 obviously covers all the topic areas, so try and cover that one as much as you can. You have what, 7 weeks? Is it 7 weeks starting tomorrow is my first session. You've got 7 weeks, and then your first exam is the 5th. So that gets you right up to that first exam. You have 7 weeks left. If you do it right, you have enough time to pick up a grade, if not 2 grades, depending on where your knowledge is. Lyra, go and check out my YouTube channel and look at my transport in plants videos. They are there for you to use. Right, Lynn, let's cover this question here, and then I'll jump between the two, as I said before. So this we're being told about a greater blue-ringed octopus. Now, the other thing to note, every time in the exam, I know this is not what the question is asking you, but every time you see something which has been given to you as the scientific name, make sure you remind yourself that that's the genus and this is the species. So they've already helped us out by giving us some clues here, capital letters, and then we've got our lowercase. It's one of the most venomous of all animals. Its bite contains a toxin, TTX. This is a neurotoxin that causes paralysis and death within minutes. Right. So they've already, again, given us a little bit of information. So this is a tetrodotoxin that is a neurotoxin. So therefore, we know that it is going to be something that can bind and affect our nervous system. So that's one of the things to make sure that when you see a question like this, you can see the hints that they've given you. What else have they told us? Right, well, they've told us that it has been discovered about the effects. Okay, it binds to the external surface of a voltage-gated sodium ion channel in the axon membrane. So straight away, they're asking, do you know why a voltage-gated sodium ion channel is important? Then they've told you that the binding of this toxin changes the tertiary structure of the channel, and this means that the channel cannot open. So just to remind yourselves, we're talking about a channel which is a protein that has a tertiary structure, so therefore is a globular protein. So again, I'm just giving myself little hints to help me to understand what's happening. So the fact that it's binding means that it has to be complementary. It's going to be complementary. Now, they've told us that it's going to bind, and they've told us that it's going to change the tertiary structure of this globular protein. So again, they've kind of told me that this is going to not bind to the active site because it's changing the tertiary structure. So it acts a bit like a non-competitive inhibitor. So be careful when you're talking about, I'm not the synaptic knob, when we're looking at that, if we were looking at a synapse here and we were looking at the postsynaptic neuron, this one isn't a voltage gated sodium channel. This has receptors for the neurotransmitter that's going to be released, the acetylcholine. So therefore, we have to be aware that we're talking here about affecting the voltage-gated sodium ion channels in the axon membrane, because that's what they told us in the question. So we should know that if the voltage-gated sodium channels open, what that will mean is that will cause a depolarization of the axon that therefore will result in the threshold value being reached. And because the threshold value is reached, that's therefore going to generate an action potential. Now, I'm not answering the question. Just to be clear, right now, what I'm doing is I'm just priming my head What do I already know about a voltage-gated sodium ion channel? Voltage-gated sodium channel opens, the sodium is going to flood in, it's going to go down its concentration gradient. And that is what is going to cause that depolarization of the axon membrane. Threshold value is going to be reached. We've got an active potential. We therefore got that impulse moving down through the neuron. So they're telling us that this TTX is going to bind. When it binds, it's going to change the tertiary structure, and therefore the channel cannot open. So why are the receptors not voltage gated? Because they are different channels have different roles to play. So these ones are the ones which are going to be detecting the stimuli and only be activated in the presence of a neurotransmitter. The voltage gated sodium channels are going to open when the voltage inside the axon becomes more positively charged, gets closer to that minus 50, minus 55, depending on which graph they give you. So it's important that you note that the receptors, when we're talking about the postsynaptic membrane, yes, there's going to be voltage-gated sodium channels there too, but the ones which are being activated because of the neurotransmitter are basically receptors that once that binding of the neurotransmitter occurs, that therefore opens the channel by changing its tertiary structure, allowing the sodium to now flood in. That then, once the threshold value has been reached, will activate your voltage-gated sodium channels to open more influx. So it's a way in which you're ensuring the filtering out of low-level stimuli, and also that they've been adapted to be specific for the role that they have to play. So remember, the dendrite leads down to the cell body and the axon leads away from the cell body. So you're thinking about the receptors which are on the dendrites. This is talking about the axon. So again, that's another bit of the key wording that you have to think about in how you can actually link it back to this. Okay, so if we carry on with the question then, so it now says, explain why this is not actually going to work properly, because they've already told you that it is not opening the channels, and because they've told you it's not opening the channels, that therefore means you should know that it's not opening the channels, therefore we're not going to be able to reach the threshold value because we're not going to depolarize the membrane. We're not going to have an action potential be reached. If an action potential isn't reached, that therefore means no neurotransmitters are going to be released. So if we've not got any neurotransmitters being released, that therefore means it's not going to pass on the impulse to anything else. So that's why this is going to cause paralysis, because it's basically stopping the impulse, the chemical messengers from being released, because the impulse can't make its way down. What question am I doing, Naveen? I am doing currently 21A. I'm actually on my YouTube channel and it should be showing you the questions live as I do them. Lynn, right, so for 23B, why can't we say carbon dioxide? Why do we have to say carbonic acid? Okay, 23B, where are you, 23B? 23B. Okay, so let's have a look at what it says. When the heart rate is too low, the level of carboxylic acid group in the blood becomes higher than normal. Well, obviously not. So when the heart rate is too low, the level of carboxylic acid group, carboxylic acid we know is where we're talking about our Q. And so therefore we know that that's not correct because we're talking about the levels in the blood become higher than normal. We're looking at that being the carbon dioxide levels becoming higher than normal because they are becoming higher than normal, that's therefore going to react with our water. And because it reacts with our water, it's therefore going to produce our our carbonic acid. So therefore, we should already know that because that is transport in animals. And we should know that because of the so why can't you say it's carbon dioxide? Because the carbon dioxide is never carried as carbon dioxide. The carbon dioxide is always going to be converted into carbonic acid. And then what you have is that carbonic acid is then going to dissociate into your hydrogen ions and your hydrogen carbonate ions. So your carbon dioxide is actually carried as hydrogen carbonate ions rather than carbon dioxide on its own, because the carbon dioxide levels of dissolving are much lower. So this is a more efficient way in which they can actually transport the carbon dioxide. So that's the reason you can't say that it's the carbon dioxide, because they're expecting you to know that that's automatically going to be converted because it's in the blood, and therefore it's going to be converted into your carbonic acid. No problem. That's the whole point in these sessions. Oh, I'm so glad you say that you're loving AIQ chat. That's what I want you to say. That's what I want people to actually really gain the information from it, because it's amazing. Could you please answer, right, 2017, 22 CII on paper one. Right, where are we going? 22 C. Hold on 1 second. Do 20. No, you didn't say 22. Where have you gone? Where have you gone? 21. 22, and you want CII. CI, okay. I, right, okay. So studies show that after damage by infection or injury, it is possible for nephron tissue to be regenerated. Adult stem cells are involved in this process. Right, okay, so when we talk about regeneration, so the first thing we've got is we're being told regeneration. So regeneration, we know for regeneration, we're going to have mitosis occur. That mitosis, then we need differentiation. Because what we're actually doing is we are replacing damaged cells. So we need that increase to occur. And then what we need is for after that increase to occur, we then need to ensure that we are having that differentiation occur. So the next thing we need to look at is, well, they've told us that they're adult stem cells. So again, they love giving you little hints, or they expect you to know this. So adult stem cells, we know, are multipotent. So just going to add that in here. Because they're multipotent, what does that mean? That means that they have the capacity to go through mitotic division and to differentiate, but not to give me any type of cell. So this is important that they're checking, do I know the difference between my pluripotent, my totipotent, and my multipotent? So that would be one of the first things that was the hint that was there, and that is a marking point. So what features of them make them suitable? Well, there we go. It's that they are multipotent, and because they're multipotent, they can become, and this is the important part, any cell type, but be careful on that one, within the tissue. And in this case, they've already told us that that's nephron tissue. No. And that's where most students actually fall down, because you talk about hemopoietic stem cells, because hemopoietic stem cells have been listed in your specification as ones that you need to know. So teachers teach it in the sense that they go, oh, you have to know about hemopoietic stem cells, but you have to have, if you damage any part of your body, you need to have repair occurring. So therefore, what you actually have is you have these multipotent stem cells, adult stem cells at different locations within the body. So for example, if we're looking at the liver and we were talking about hepatocytes, the liver is one of the organs that can regenerate at a faster rate. And therefore, that's because they have multipotent stem cells that have the capacity to go through mitosis and differentiate into the cells needed to regenerate. So the minute you see a question that says regeneration can occur, that therefore means that you are looking at a multipotent stem cell because it's an adult stem cell. What topic would that be? So when we're talking about potency and stem cells, that's module 2, 2.6, but this question is actually linking it to the excretion module 5 because it's paper 3, and that's where they can do that interlinking. So yeah, does that help? It is important. Those are the sorts of things, like figuring out what you don't know is really important, because when you know what you don't know, i.e. like that little bit of information, the next time they ask a question, it will help you. Thank you. Do you know what it is? It's not about being smart. It's a funny one. I'm going to go on a bit of a rant on this one. It's not about being smart. It's about the fact that I have specialized. I would say I'm an expert on OCRA because I've specialized. I took 6 months to really hone in on what the exam board wanted you to know to build my YouTube channel. Now, I literally live OCRA in and out with all the clients that I have, all the students that I work with. I do teach the other exam boards and I have to adapt to their questions, but I know OCRA and I know what they're looking for. But it's about the fact that you've got to remember, I have 20 years of experience at teaching this. Well, I'm repeating this every year, right? I'm repeating this again and again and again, but I forget things. After the holidays, the summer holidays, I have to go back over and relearn it all. I basically built AIQ Chat for myself and my YouTube channel for myself too, so that when I'm like, oh, I'm so tired, I don't have the time to just quickly read the textbook, I can just have a quick check of my videos, but now I remember what I said then. Now I remember that. So it's about repetition. It is, it's like living OCR is like completely stuck in my head now. You are very welcome. And if you want to, I am on my YouTube channel with some questions live. And if you have questions you want me to cover, please, people are asking away. Question 14 on paper 2, 2017. Yeah, give me 1 second. And the reason I'm doing it on my YouTube channel is because then it's there for you. So what do we want? We want 2017 paper 2. 2017 paper 2. Nope, I didn't want to do that. That's not what I want to do. Give me a second. I opened it in the wrong place. I want to open it here and then open this. And you want me to do question 14. Right, 2017, question 14. Here we go. This one here. Lynn, is that the one you're looking for? Just to confirm that I've got the right one. Yeah, cool. Right. So this one's an interesting one because it's asking you about the transmission between animals and plants. So we've got the 3 methods of these pathogens being transmitted. Now, this is the bit you really need to focus on because students skim read. They skim read all the time. And therefore, because they skim read, they may miss a part of the question that says, which of the methods of pathogen transmission does not apply to plants? So which of these does not apply to plants? So what do we know? Well, we're talking about the plants, and if we're looking at direct contact, can plants have pathogens passed and transmitted between the plants through direct contact? Yes, we will after that one. So direct contact. So this is where, for example, infected leaves touch other leaves. So if we have infected leaves that are going to touch other leaves, that is therefore going to be direct contact. So which of the methods of transmission does not apply to plants? So one does apply to plants because we have direct contact. So in this case, we automatically therefore know that direct contact does apply. That is a not question. So anyone that has one in it, therefore would remove those, right? So you're saying, isn't 3 the only wrong one? In this case, why is it the wrong one? So it's really interesting that the plants can touch leaves. So it's not that that's wrong, it's that one is correct, and therefore we need to remove C. And that's the bit where students get confused at. They think that they are answering it, but actually what they're doing is they're eliminating the wrong answers. So when they do the elimination process, you have to take the time and think about which ones are actually the ones that are correct for the statement. So one is a correct one, that the direct contact does happen and does apply to the plants. So therefore, that is a correct statement. The vectors, how do vectors pass on the pathogen? Well, a vector is things like wind, things like water. So vectors can transmit pathogens to plants. So therefore, exactly. So vectors are also things that can happen and can apply to plants. So in this case, now we know that it has to be at least 1 and 2, and therefore it cannot be only 1. So we've now eliminated the C and D, and Amna, you're right in what you said. The droplets, when we talk about droplets, we're talking about sneezing, coughing. So therefore, which of these does not apply to the plant? This does not apply to plants. So we remove number 3, and therefore leaving us with the only right answer, which is B. So it is really important when you're going through these questions and you're practicing them, that you're not just checking the marks game, that you're actually writing notes to yourself on how do they actually work? How is it that direct contact occurs between the plants, leaves touching each other, the wind, the water, the soil, for example, and then why the droplets was the incorrect one, because that's going to make a big difference to you getting it. No, I know. Do you know what, Paul? I was going to say that was literally something that I was then going to say as well. That it's so confusing sometimes for students in the fact that if you think about a droplet, are you talking about it being transported in water, or are you talking about it in the case of the coughing and sneezing? And in this case, they were referring to it with the coughing and sneezing. And so because of that, anyone who thought like you did about the moisture droplet would just not get the answer. If enough students gave the answer that said that that was true, what would have happened is the examiners would have got together and argued and said there's enough people who are actually saying this that we should actually allow it, because technically it could be a right answer. And this is why sometimes when you look at a mark scheme, it will say one answer, and then after that for the multiple choice, it'll say, oh, but allow this one as well. And that's because the examiners who marked it at that point in that year, they made the decision at that point that that was the answers they were going to accept, because there was enough students who gave that those answers, and therefore that is why they mark schemes may seem very specific one year, students memorize them, and then the next year they don't get the same answers or get credited for the same answer. So it's really important whenever you are looking at these mark schemes that you do know that there is deviation each year group. Right, Amna, can I do question 9 on, hold on, 2017 paper 2? That was this one, wasn't it? And you want question 9. Let's jump back up. Where are you? Where are you? Here we go. Okay, so is this the one you want? Question 9 and then 17C on the same paper. Yeah, right, okay. So hormone HCG can be detected in, oh, I hate this question. I'm not gonna lie, I actually hate this question because it is a interesting one where the people who wrote this question, I don't know why they decided to write it this way. It's really not a very nice question. Oh, hold on. So have I done the wrong one? Which one am I on? This one's 2017 paper 2. Question 9. Oh, there we go. This one. Yeah. Okay. So that other question is quite a horrible question. And yeah, it's one of those ones that sometimes you just have to know the answer for by eliminating the other ones. So that's the one thing I would say. Sometimes it's just you have to just know how to do elimination to get you back. But anyway, let's look at this one. Cut pieces of agar jelly could be used to investigate the factors affecting diffusion rates in cells. Now, straight away, they've said diffusion rate. I know they're going to probably talk about my surface area to volume ratio. I'm going to just give myself a little hint because they're talking about diffusion rate. So we're either talking about the surface area to volume ratio, or we're talking about the diffusion pathway. 4 pieces of agar jelly contain universal indicator with soaps in the same concentration, right? So, and this is, someone kept saying to me about how do you do practice of the practical questions? Well, this is a practical question actually being thrown in there for you, because what you have is they have controlled the same concentration of my hydrochloric acid. They've also done it for 1 minute. So they're giving you little hints that they are controlling the time, they're controlling the concentration of the hydrochloric acid. They've used an indicator. Why have they used an indicator? And I know I'm not answering the question. I'm just trying to help you to see where the practical skills are there. They've used an universal indicator because that's going to give you a color for you to assess. And therefore, we're looking at qualitative data here because we're looking at that color changing. So then it says, right, the cubes were then removed and blotted dry. So they're going to be removed, they're going to be blotted dry. Which of the following pieces of agar jelly would be the first to turn entirely red? Right, so hydrochloric acid, they've told us in the question that it is acid. So therefore we know if we're looking at our pH scale, it goes from 0 to 14, 7 in the middle. We've got our red color down here. We've got our purple color here. We've got our green color here. So therefore, they're expecting you to use GCSE knowledge to know that this is acidic, this is alkaline. And if I'm using a universal indicator and I'm using hydrochloric acid, that is therefore going to be affecting, let me just, this second, the points. Now, remember, with the surface area, it's going to be hitting every single surface at the same time. So therefore, what are we looking at? We're looking at the movement of it coming in from all sides. So what have they given us? A cube with edges 4 centimeters each, a cuboid with edges 2, 4, and 6, and a cuboid with edges 3, 3, and 5, or a sphere with a diameter of 4. So they've already told us that cut pieces of agar jelly are going to be used. 4 pieces of agar jelly containing the universal indicator were soaked. The cubes were then removed and blotted dry, which of the following. So we've got the fact that they're cubes, the fact that they are blotted dry. Which of the following pieces of agar jelly would be the first to turn entirely red? So we know that it's not going to be a sphere. So we can remove that from the answer. So we know it's not going to be a sphere. So now we need to think of, right, okay, is it going to be a cube with edges of 4 centimeters each? Or is it going to be a cuboid? And is that cuboid going to have 2, 4, and 6, or 3, 3, and 5? So we're talking about this agar jelly. We're talking about hydrochloric acid. We're talking about how it's going to get in. And so therefore, it's going to diffuse at different rates due to the way it's going to be interacting with the hydrochloric acid. So I've got to think this one through. And so if I was thinking about this and I was trying to come up with an answer to it, I've got to think about, I have 4 pieces which I have placed in my universal indicator. And because I've placed it in my universal indicator, what's going to happen is I said it's going to hit all surfaces. Well, if one of those surfaces is actually going to be at the bottom, that's going to affect the rate at which it's moving in. So therefore, I would assume that it's not going to be my cube with edges of 4 centimeters by 4. I've got 4 by 4 by 4. It's not going to be a cube. And so therefore, if we do this for my 4 by 4 by 4, and then my 4 by 4 by 6, I've done that the completely wrong way around. Let's try that, switching those two the other way around, because it's surface area first. So Amna, you said you've worked out your ratios. What's your ratios? What's the 4 by 4? The surface area to volume ratio. See, I'm like, where's my calculator? And here it is here. Right, so 1.5 to 1 is that one. What's the second one? B is 1.58 to 1. And then what's C? 1.73 to 1. But, okay, so although your surface area to volume ratio is higher with a cuboid that's edges are 3 by 3, you've got to remember that it's asking about which would be the first to turn entirely red. So therefore, it would be the one that's got the smallest at one side. So you've got your 2 by 4 by 6, 3 by 3 by 5. That's a horrible question. So the correct answer is B, but I'm going to have to sit and think about why exactly that. I would say because you've got the smaller one, but if you're saying the cuboid has the higher edges, let me just see. Let me go over here. See, and this is where there are questions that actually, for some of us, makes us go, as teachers, it literally stumps us. And then we think to ourselves, well, how would we answer this if we know If we know the question and we know the mark scheme, then we should know the answer to this. But actually, I'm now sitting here going, this is a bit of a brain teaser. Let me think about this. Let me try and work this out. Because actually, I'm like sitting here thinking, well, it's a smaller size on the edges. Yeah, well, that's what I'm thinking. But if it's got a different surface area to volume ratio, then it's like, why have they given that as the question if that's the answer they want? There's obviously something not right. What do I want? I'm trying to find the question. Oh, I can't spell. Well, this is the thing. It does actually make you stop and think and go, oh, well, hold on a minute. Have I done something that's not right? So what we're doing, 2017, right? Maybe it's multiple choice. I want to see what the examiner's report said. No. It was paper 2 we're doing, isn't it? Only 1 in 5 candidates achieve the mark on that one. Well, yeah, that's why I'm questioning it, because they're saying the answer is B. And so therefore, I'm trying to think why the answer is B on that one, because you would expect it to be the one with the highest surface area to volume ratio, like Paul agreed. So what am I looking for? Calculator. If you've done your calculations correct, so what have we got? A cuboid with edges, 3 by 3 by 5. So if it's 3 by 3 times 2, and then you've got, adding with that, your 5 by 3, 5 by 3, those brackets, times 2. Oh, hold on, I did that completely wrong. So 5 by 3 by 2. So I got my 48, and then, oh, okay. So if you've got your 2 by 4, so that's my a cuboid. So I've got my 2 by 4 by 6. So I've got 2 by 4 times 2. And then I've got my 2 and my 4 by, oh crumbs, I'm just writing everything down here now, aren't I? My 4 by 6 times 2, and I add them together. And then obviously I do my volume, which is 2 by 4 by 6. So what's the number it gets? So 2 by 4 times 2. So Amna, what have you worked it out to be now? I need a piece of paper. I've got my computer screen. There we go. That's the reason being. So yeah, what you've got, that makes more sense. 1.83 to 1, which is why B is the right answer. Yeah, that was a confusing one for me, because I was thinking, but again, it's really important. That's one of the things I would say to you. It's really important when you're doing these questions that you do take the time to practice the maths and double check the maths to make sure you have put in the numbers correctly, because that will make a difference of you getting the mark or not. But yeah, you even threw me off on that one. I thought I was going insane. Okay, where are we at? 17C, since I'm on this paper. Was the 17C on this one too? 17 C, where are you? 17C on 2017. Yes, right, okay. So I love this one. This is a good question. It's always the way, and I do as well. I hate the math questions. They take me so much longer. It's like the minute I get a math question and I see numbers, I can see them in front of me and I know roughly what to do with them. But sometimes when I start to do it, I get confused and then it's like it just goes awry and I get it all wrong. And so therefore I have to do it again and again and again. Now, this is one of the things, if I was doing this in a session that was planned, I would have gone through this so much on my own to make sure that I knew how to do the answer, because that's what I do. I find the ones that I struggle with and I then make sure that I can answer it. If I get stuck, I then say to Alan, I'm like, can you just help me out here? And as I'm trying to explain it to him, he will then try to give me an answer. And I'm like, oh, now I know it. I was like, I don't need you anymore. But I just needed to talk it through with somebody. There we go. And so because of that, that's sometimes a good way for you to improve your math skills, but there are always going to be questions that throw you off. And that's okay. There's always going to be questions that make you question, oh, I'm not very good at this. And that's okay as well. And there's questions that like I do. And when you get stressed, you could just go, "Oh, stuff this, I don't know. Right, I'm going to finish now and off I go." But actually, it's really important to just say, right, I'm not good at that. Maybe I spend a bit more time on that sort of question so that I get better at it. So if they do throw a question in at me, but don't waste too much time on it if there is other content that you don't know, because it could be the difference between you gaining 1 mark or not. That's a 1 marker. I would have attempted it, got a bit frustrated and bothered, moved on. If I had some time later on, I would have come back. Otherwise, I would have just guessed it and see it, hopefully got it right. For this one, could we talk about repressor proteins being produced by recessive alleles, which bind to operator region on dominant allele? Right. Well, let's have a look at what they've already told us. So they've told us that the Y, capital Y, so dominant production causes the production of an enzyme that breaks down the chlorophyll. The recessive allele Y is a mutation in the Y allele. The Y allele codes for an inactive form of this enzyme. So again, straight away, I am going to just remind myself about what this is saying. What is an enzyme? An enzyme is a globular protein. Globular protein, which has a tertiary structure. So straight away, I'm just writing some keywords down, and then I'm going to see if I can apply them to the question that's in front of me, because that's how you actually manage to gain the marking points, by writing those keywords and then applying it to the question that's in front of you. So in this case, what are they actually asking me to do? So outline how the dominant allele, because they've said the capital Y allele, codes for production of the enzyme. Haha, it's not production of the enzyme, it's the production of the globular protein. So therefore, production is transcription and translation. So there's the first part of this question. So the first part is, number one, the coding, genetic code. The second part is protein synthesis. And then explain why the lowercase enzyme gives you a different primary structure. That therefore, we're looking already that we've seen it's a mutation. That third part to the question is looking at mutation. And then, yes, you could talk about repressor protein, potentially. Um, Yeah, it is real. Um, oh, thank you. Thank you. That's really lovely. The first two things. So in this case, hold on, let me have a look at what it's actually looking for you to say. So a reference to the nature of the genetic code and an outline of how alleles are transcribed and translated, and a detailed explanation of why the Y allele results in a different primary. And the reason it doesn't allow that is because it's talking about a different primary structure. So therefore, your primary structure is your sequence of amino acids. So because you're talking about the repressor protein is the one which is going to stop transcription. That's the role that it could play. So that would be a role of the protein from the lowercase y. That's not the fact that it's coding for a different primary structure. So although you have given a role that it's played, that would just be a ignore information. You're not incorrect in what you're saying. However, you haven't answered the question. So it's about ensuring that you are answering the question that's been asked in front of you. So the fact that it's a mutation, what they're looking for you to really talk about is that mutation that they told you in the question. I'm going to circle it again. The mutation that therefore is resulting in this different primary structure. And that different primary structure is because you have a different base sequence, that genetic code, that's going to have a different codon, different anticodon. So if, for example, we had ATC in our DNA, we know that that would give us a codon, which would be UAG. That would therefore match the anticodon, which would be AUC for the anticodon. If I just write this one being the codon, and this one being the, what was this one, the DNA. So therefore, if instead of ATC, I now have AAC in my DNA strand because I've got a base substitution, I now have UUG as my codon. I therefore now have AAG as, no, I don't, AAC even. Let's try that again. AAC as my anticodon. So that's what they're looking for you to be able to talk about. You are very welcome. Yeah, so it's actually, so the recessive allele, you're right, is not coding for a protein that breaks down the chlorophyll. It's actually preventing the breakdown of the chlorophyll, which therefore keeps it as the primary pigment, which is why it's green. Absolutely right. Now, for 18A, hold on, what was that? Why can't we say thickening of wall on this paper? 18A. This one. Elle, is this the one you're looking for? And then 18B II. So hold on, 18B II. Right. State how a clone of potatoes could be produced for this investigation and explain why it's important to carry out this procedure under aseptic techniques. Also, why can't we mention pathogens? Or hold on, 18B, have I missed something? 18B II. Why are you mentioning pathogens? We're talking about a resistant strain. We're talking about the infection. Let me just go back up and read exactly what it says. So potato plant, staple food plant in the world are susceptible to infection by a pathogen, which causes a disease known as potato late blight. So Okay, so when we talk about this one, it's an interesting one because they are expecting you to note the fact that you've got potato late blight. So this is one of the infections that you have to know about that is caused in the plant. So what actually causes the potato late blight is the pathogen, which is the Protoctista. So that's the first thing to note, that we're looking at a Protoctista or protist pathogen, just as a backup of our extra ones. So then they said some varieties of potato are resistant. Now, I do feel like resistance is coming up this year. I don't know why. I feel like there's something. How do you get a resistance? The resistance would be that there is some form of a mutation. That mutation could result in the production of, let's say, a protein, which is going to stop the pathogen, whatever the pathogen is. So I'm just going to add that in there just as a bit of a reminder. Okay, so some varieties are resistant to the infections. State 2 ways in which an individual plant could respond. Okay, so this is basically calling up your knowledge on plant responses. So module 5, and also defense mechanisms, which is module 4. Hold on one second. Potato leaf blight. Hold on, now you're questioning me, and I'm like, hold on, is that not the Protoctistin, and therefore the black Blaxicatoca is the fungal infection? Oh, put me on the spot here. You might be right. I have to say there are bits where I get it wrong, so I'm just going to double check that one, because I don't want to give the wrong information. Nope, I am right. Potato late blight, according to OCR A, is Protoctista. Fungal infection for plants that they need to know about is the Blaxicatoca. Look at me go. This is the thing, I'm like, I know it, I should know it, but it's been a while since I've done it, so I do like question myself, but no, it's Protoctistin pathogen for the potato late blight. But thank you for questioning me, because actually that's a good thing, that sometimes as teachers we make mistakes, and it's okay for you to question us, to ask to double check, and for us to go and double check that we have got it right. And that's the thing, that different exam boards will do things in different ways, and some of us have expertise in that specific exam board, but we can still make mistakes. But actually in this one I am correct. Oh, see, this is an interesting one. So now you're saying that the before infestatia, so it could be that the exam board have literally just thrown a name of one out there. Now I'm going to double check this because I don't know the names of my this is where my flora infestans. Be careful. It is a protoctistin. It's a fungus-like microorganism. It's not a fungus. So it is a fungus-like infection, but it's actually a protoctistin, which is what causes it. And it's one they have to know for their exam. Anyway, back to this question. Sorry, Amna, you asked... sorry, you asked about why can't we say thickening of the cell wall on this paper? So when you're saying the thickening of the cell wall, what are you talking about? Because if you're talking about the cell wall, so this is where you have to be careful with your wording. Your cell wall is made of cellulose. Whereas when we talk about defenses of the plant, we're talking about callose, which is a different kind, oh crumbs, can't even spell polysaccharide, of polysaccharide. So be careful. The cell wall, you know, is made up of cellulose. If you had quantified your answer and said that it is the thickening with lignin, then you would get the marking point. So it's about ensuring that you are giving the right information to no, the cellulose doesn't thicken. It is the lignin that is there to create a barrier around the cell wall. So the cell wall is made up of the cellulose. Just to add in, the cellulose is obviously our beta glucose molecules. So another thing to note, 1 to 4 glycosidic bonds, also 180-degree inversion. So I would just be writing a few little notes to myself just to keep that in the forefront of my mind because I'm revising it. How do I tackle suggest questions? Again, it depends on the question. I have to take the time to look at what's in front of me. So let's just have a look at 18BII. 18BII. So the other thing I would say to you is when you've been given a question that looks like this, so resistance of different varieties, infection, they've told us they've got 3 clones. They've told us that they're grown in adjacent fields over the same time. So I'd be saying to myself, right, what are my control variables? For example, what are my sample size? I'll be looking at a sample size. I'll be saying to myself, have they actually done any repeats? Before I move on to that question, I'm looking for notes that they've given me because they've always, that's the one thing I would say OCR are very good at doing is giving you hints. Their questions are not straightforward, but the hints are there. Excuse me, so you just have to find where those hints are. So looking at this, they've told us it's the percentage of the area of a leaf with visible infection. I can see, and I'm just going to note this because this is something you should have a look at in the exam. Each time, oh no, I didn't want to do that. Let me try that again. Each time, have they done the same number of samples? So that's another thing to look at. Each time, are they doing the same number of samples taken? Because what you'll find when I look at my sample size, if there's some questions practical based, they may have only done, let's say, 5 samples taken in A, 10 in B and C, and therefore are the results valid, etc. Is something to consider. And what was the question? You said and I will finish on this one. Suggest how a clone of potatoes could be produced. Right, we're talking about cloning. So if we're talking about cloning, we need to consider the fact that the clone has to be asexual reproduction. It's going to be through the process of mitosis. So again, I'm using their words to prime myself on what might be that they are asking me in this question. So how am I going to clone this? Well, if I want to clone it, and I'm cloning a potato, I'm probably going to have to do my tissue culture. So I want to do tissue culture where I'm going to remove a small sample, which is called an explant. Now, the explant needs to be removed, and that explant being removed needs to ensure that there's no contamination. So asepsis is about ensuring no contamination. So I do think that that's potentially coming up in more detail in your exam this year. Guys, I am going to finish now because my phone has gone to my night mode, which tells me it's time for me to go to my bed. So thank you for all of you who have joined me tonight. And for those of you watching, thank you. Please do comment if you have questions on my videos. I will keep doing as much as I can. When I can, obviously, I prioritize my paid tuition sessions first. So I jump on here whenever I get a moment, especially with everything that is going forward with your exam, because you do only have 7 weeks left, I will try and come on as much as I can. I can't guarantee it will be like this, but I do this normally a Monday and a Wednesday. So I will be back on Wednesday again to help. And so yeah, thanks for joining me, and join me again on Wednesday at about 8:30. Bye guys. Don't worry about it. Don't worry about it, Paul. It's a case of there's always bits that we don't know. That's the key thing. And it keeps me on my toes.